And our first speaker today, not Dr. Michael Klein, but Cassie Sickhorse. I just learned to pronounce her name. So she's a community outreach specialist. Sorry, she's from Washington State Department of Ag, Asian Giant Hornet Community Outreach and Educational Specialist. And she also has a very good blend of academic career in natural resources, environmental science and education. Cassie, okay, you can take away from me now.
Well, hello, everyone. I'm over here in Washington, and I'm going to talk to you about Asian giant hornet. Now, I do work from home, so my bandwidth isn't that strong. If you're having any trouble at any time hearing me, I do have like a couple quick short videos I'll play. It might help to turn off your video if that's coming through kind of fuzzy for you. So I'm going to share my screen here. You're going to see your faces momentarily before I start my slideshow. Okay, so if someone can just confirm for me that you see one slide and it looks good and ready to go.
Yes.
All right, so Asian giant hornet, right? We've all heard that infamous murder hornet. Well, it also has other nicknames like giant sparrow bee and yak killer hornet. Just a really quick background on that murder hornet that you all are probably familiar with. Basically, Asian giant hornet was discovered in Washington in December of 2019. So the New York Times published an article, Asian giant hornet threatens honeybees in the Pacific Northwest. And it did gain a little ground and people took interest in it. So they did a follow-up article in May. They interviewed some people here at the agency, interviewed people overseas, and one gentleman happened to say, well, I call it the murder hornet. So they published their next article with that in the title, and since then we have just trucked along. So what is the Asian giant hornet? Well, it is the largest hornet in the world, and adults range between 1.5 inches to 2 inches in length, right? There's no such thing as a baby Asian giant hornet. When they emerge from the nest, they are their full adult size. Okay, a couple characteristics to distinguish this from other species, right? The big red circle I have over its head for a reason. It's very large. It's orange and yellow in color, and it's prominent, okay? It's as big and as wide as its body. This is gonna set it apart from a lot of other species you might see. And then we got the distinct orange-yellow band striping on the abdomen. Now these guys and gals, mostly gals, like subtropic to moderate temperatures, hence their ability, to come from their native range and attempt to establish over here in the Pacific Northwest. Okay, I already told you that they were confirmed here in December 2019. However, before that, in Nanaimo, British Columbia, they did eradicate, the beekeepers got together and eradicated a nest in September 2019. Here in Washington, Asian giant hornet threatens honeybees, human health and agriculture specifically, and I'm going to detail those throughout this conference for you. So moving on, I'm going to tell you a little bit about Asian giant hornet, a little bit about the background on them, their life cycle and nesting. One, because physical safety is important, and that will help you understand why the agency, why we do certain things like trapping and reporting at certain times of the year. So Asian giant hornets are typically non-aggressive unless they feel threatened, okay? They're an apex predator, meaning they're top of the food chain, nothing really bugs them. So they don't really care about people until we get to the nest. This is why it's important to know about the nest, okay? They're actually typically ground nesters. What they'll do is they'll find a decayed tree cavity, like at the base where the roots have rotted out, or potentially an animal or rodent burrow, okay? And they will actually they will make one entrance into the nest and then continue to harvest and dig that out. So you won't see the nest. You only see evidence of hornets coming and going, which I'll show you in the next video. Now these nests can be over 2 feet wide. They'll have multiple combs like you see in the picture here, and they will produce hundreds of adult hornets. Now we have found 2 nests here in Washington, and those have actually been up inside a tree cavity. In their native range, this is typically very rare. So I can't tell you if that's how they're going to continue to nest here in Washington. It's the first time they've been outside of their native range. Also in this picture here, you see the combs, they're laid out for you to view. They actually build those combs by stacking them together as they nest. So here's a video. Remember I said you wouldn't typically see the nests. You only see evidence of them coming and going. So this is why it's important to, if you're outside, to stay on trails and be safe. so you don't accidentally encounter an Asian giant hornet nest. Okay, the next thing I'm going to talk to you about is their life cycle, right? This helps you understand when we trap and a little bit more about the hornet species. And then I'm going to actually pause after that in case you guys have any questions, comments, and then we'll move on again. So take a minute looking at this graphics. We're going to start in the top and move in a clockwise position. So in the winter, the males and the workers, they die, as well as a founding queen that made that nest. The only thing that moves forward are new queens, okay? And they overwinter, meaning they find somewhere sheltered, potentially dig themselves in the soil. And then in the spring, they will emerge. And if they successfully made it, they will attempt to begin to build that underground nest. Okay, then what comes after spring, guys? What's the next season? Summer, right? So as we get into summer, as that begins, workers will start to emerge. She begins and only lays the workers. Those are the ones, the gals that we'll see. They're out flying around foraging. They eat beetles, bees, wasps, dragonflies, moths, butterflies. They take those insects back to feed their young, okay? And they do this primarily throughout all of the summer and into the fall. However, then when we begin to reach that fall, that colony reaches its maturity point, meaning that queen that started the nest is now going to switch from laying workers to laying new virgin queens and males, okay? Now we're in that fall time, remember, this is in the reproductive stage. Those queens and males, right, are gonna emerge from the nest. They mate really close to the nest. Then the new queens will fly off to overwinter. Those males will die. The workers are all going to be dead, right, as well as that founding queen. And then moving forth, that cycle will repeat itself, okay? Now, real quick before I move on, you see in this graphic, it gets red over in that fall time. Well, for a reason, because that's when they start to reproduce things that could go on to next year. And also while she's making these queens and males, that colony needs more protein and food in their diet at that time. And what do y'all think is a great one-stop shop source for a lot of food? Any guesses? Beehives. Okay, so before I go into talking about how they potentially decimate beehives, if you guys have any questions, go ahead and put them in the chat box, or if you're capable of, I don't know if we unmute ourselves, but I'll pause for a second in case there's questions before I move on. Okay, seeing no questions, okay. If Asian giant hornet were to arrive in Hawaii, would they skip the overwintering process? So in their native range, they like the subtropic to moderate temperatures, right? There's a climatic study published, and I can't remember where Hawaii falls. They're not going to typically skip the overwintering process, right? Because basically what the queen is a solitary queen that sits inside of the nest. Okay, so they mate, they get fat, they go off and start their own nest, depending on, they've never been in Hawaii, so it'd be hard to tell. Depending on that time of year, if it's right, I guess they could potentially go into starting their nests, but I haven't read any research on them not doing that. So I actually don't know the exact answer to that question, but once they leave the nest, they have the potential to start a new nest. Yes, it has the wet, dry climate. I know that. I'm just trying to think about, usually I get asked if we have a really rough winter, will it stop the hornets from moving forth? And that is typically a no. So, and I can actually, at the end of this, I'll go ahead and publish the link to that study so you can look at the map and see where it sits. All right. Resuming sharing. So we were talking, I was talking about then that, what time of year is that they get really hungry? Does anyone remember? It's that fall time, okay? And other insects, you know, they aren't reproducing and getting ready for winter like the bees are. Okay, in the fall time, the honeybees are starting to lay their winter bees. So it's a really good time for the hornets to stop in and get lots of protein. What happens is the Asian giant hornets will mark a honeybee hive with a pheromone, right? And they might not go right away. They might wait a week or two until they are ready to decimate this hive. What happens is that marking of the pheromone signals other hornets to come here. Once they get to the honeybee hive, the adult honeybees fly out to protect themselves, and then they begin to chop their heads off and drop the bodies. Once all the adult bees are decapitated, they will enter the hive and harvest all of the brood and the pupae from the honeybee hive, taking it back to feed their young, okay? And they will do this until the food source is no longer good. They don't want it, or it's all completely gone. Typically, that's the answer. Okay, remember this typically happens in the fall. Okay, here's a statistic from their native range. So if I have 1 to 20 Asian giant hornets, right, how long do you think it will take them to wipe out a beehive? And that is typically 45 to 65,000 bees. Any guesses on how long that takes?
Jason.
Join the meeting.
It's about 90 minutes, okay? So obviously if these guys establish, honeybees go down, and you all know that's gonna impact our pollinators even more than they already are, agriculture, etc. Okay, 3 days, a little bit too long. Okay, so then people ask, well, how do Asian giant hornets and honeybees survive in their native range? Well, a couple differences. Over here in the States, you know, we have European honeybees, okay? Over there, they have Japanese honeybees. Couple differences, ours are really good at producing honey. Theirs, you know, they still do a good job, but not as strong, okay? The Japanese honeybees do have a defense mechanism against the Asian giant hornets. If the Asian giant hornet attempts to enter and mark that hive, the honeybees will fly and form a bee ball around it, buzzing and warming their body temperatures up just enough to essentially cook that hornet alive, and it drops and dies, okay? Our honeybees don't have that defense mechanism, and for them to learn that would take a very, very, very long time. So why don't we import Japanese honeybees over here? Well, Japanese honeybees have a mite called the Tropolaelaps mite, and compared to the mites we have here, it's going to look like the Hulk Hogan of mites. Mites are kind of this thing that will make honeybees sick. It'll make the hive weak. It's really hard to treat for mites, and a lot of the times, or sometimes you just lose your entire honeybee hive. So that's kind of the wrap up on honeybees. And the last thing I'm going to tell you about the Asian giant hornet background, maybe if I can go to my next screen, is a little bit about the history in their native range. So way back when, Asian giant hornets served a purpose. They were a great source of protein. Now that has kind of equated into them actually being a delicacy. Remember, they're typically non-aggressive, but if you come upon one of those hives, they get a little mad. So people actually go and harvest the hives and take the brood and the, sorry, not hives, they harvest the colonies, taking the brood and the pupae of the Asian giant hornets, then they fry it up and have a feast. It equates to being about $31 a pound in our money, okay? They also will put Asian giant hornets in energy drinks as a novelty using their spit. They'll eat the adults, and kind of like people do the tequila and the worm, they will do the hornet and the alcohol. So that's kind of it I want to share with you about Asian giant hornet. And then I'm going to go into telling you about our program here, what people do to help, and what the state agency does to help. So before I move on, do you guys have any questions for me? Are Japanese honeybees just a different species or a variety of Apis mellifera? They are so they're not a different species. They're kind of like the subvariety, but the idea of them hybridizing could potentially happen, but they don't live in the proximity of each other. So them hybridizing is like unlikely. Do the Asian giant hornets also attack solitary bee nests? My answer would be yes. We were able to actually analyze some of the nest remnants from last year, and they were attacking things like paper wasp nests, yellow jackets, bald-faced hornets. I don't believe there was any mason bees found because that's kind of a different time of year. As well as this year, what we've been seeing is they have been on different various wasp nests, raiding them as well. All right. Okay, I am going to move on here. So Asian giant hornet in the United States has only been detected in Washington state, okay? And it's pretty much just the northwest corner of Washington state. So this is a map here of what the state looks like. And all these orange and red spots are where people have reported sightings or potentially caught something in a trap. Anything that's orange on this map is unconfirmed, meaning someone sent a picture that was not an Asian giant hornet, turned in a specimen that was not an Asian giant hornet, or unfortunately there wasn't enough information to go with that report, so it stays unconfirmed. Any red circle or any red diamond you see are our confirmed sightings. Last year we had 31 detections. This year we've had 3. Last year we removed 1 nest, and this year we removed 1 nest. Now on the map, you see a blue dot here. That was also an Asian giant hornet that was found further down south in a different county. It was also a different introduction in a different species, kind of a weird time of year. It might have potentially been a carcass that fell out of something that was being unpacked. Okay, then across the border, because I'm very close to Canada or British Columbia, they've had 6 reports, all kind of in that same range between the years 2019 and 2020, nothing yet this year. And then they had the 1 nest that was removed in 2019. So basically what this map does for us is it tells us where we need to place our traps. So take one of those red dots, okay? And I'm going to show them to you blown up here on this next map. This is how an integrated pest program, this is how a response works, okay? So one of those red dots is where a hornet was detected. From that, we're gonna basically go out in every direction, 8 kilometers, which is about 5 miles. And then we're gonna basically draw a circumference around that, okay? That's gonna tell us the areas that the Asian giant hornets may be going, because they only fly about 8 kilometers from their nest to find food before they go home. Remind you, we don't know where the nests are yet. That's our goal, to find them. Then we just overlay the grid pattern that you see there. That will tell us how many traps to place and where, okay? But it's not just us trapping here in Washington. We've reached out to the citizens of Washington, and we have asked them, if you have the time and you are able to deploy an Asian giant hornet trap, please do. And citizens in Washington last year responded awesomely. They placed 1,500 traps, and they responded this year too, and their numbers are about 700 right now. So this pest management program really is integrated between agency and citizens, as well as citizens reporting, that leads us to find the hornets, to find the nests. So what these traps look like, you're gonna see in this bottom left picture here. It's a clear plastic bottle, like a 2-liter soda bottle, an orange juice bottle. And then ironically, it has orange juice and rice cooking wine in it, okay?
Wait, Cassie.
Yeah.
It looks like we cannot see your slides, which you are.
Can you see that?
Okay. Yes, now yes.
You guys,
yes.
Did y'all get to see this one?
Mm-hmm. Yeah.
I don't think you saw this one either. Anyway, this is that grid I was talking about. We got 8 kilometers from there. That tells us where to trap. And here's what the traps look like. Okay, so that mixture in that bottle is orange juice and rice cooking wine, okay? It gets hung up from a tree. It's a carbohydrate, sugary sweetness, little bit of fuel for the hornets while they're hunting. So if they fly by and smell the scent of that bait, they're essentially lured into that trap, okay? And that is how we trap for Asian giant hornets. Sorry, guys. Okay, so, right, if we have an Asian giant hornet in that trap, well, that's good because that gives us the detection point, but we can't do much with a dead Asian giant hornet. We can research it, but we really need a live one to help us find the nest. So once we catch a hornet or we have a confirmed detection, we go out and we place a bunch of live traps or modified traps. Same thing, the hornet gets attracted into the trap, but it doesn't drown and die. Or sometimes people report Asian giant hornets to us, and then we're able to go out to that property and potentially just net one, okay? And then we have a live Asian giant hornet. Anybody have any idea why we want a live one? Right? Because I mean, obviously we don't want them here, so having them dead is fine. All right, so I see a question here I'll go back to, but the reason why we want a live one is because we have radio tag transmitters. We attach these to the hornets. By nature, when the hornet is let go, she returns back to her nest. So by attaching that tracker, letting her go, we're able to follow that signal back to the nest to remove the entire nest, because the only way to get rid of these guys is to remove the nest, okay? Now, I will share a couple links with you at the end with the information on trapping, etc. I do have to say that if you're not in Washington, we do not recommend you to place these traps. The traps have little openings on them. There was also another question here about getting a lot of non-target species in these traps. There is, I don't know if you can see, there's a little tab sticking out here. So there's 3 entrances in the top of the traps where the hornets fly in, okay? Basically what happened last year is when the citizens were helping us trap as well as we were trapping, we actually had everyone mail back every insect they ever caught in the trap, as well as we analyzed them to make sure we weren't harming any of our beneficial pollinators. They do attract other wasps, like bald-faced hornets and yellow jackets that we have here, but no other honeybees, bumblebees, or other beneficial species was harmed in us deploying these traps. Okay, so I'll explain a little bit more about this transmitter that your guys are seeing here, because there was a question in the chat about it. Basically, once we have the live hornet, we chill her out on a little bit of ice, okay? To make her a little less mobile, we take Kevlar thread, glue it to the tracker, tie around its waist, let it warm up, she eats and flies off. Then the trackers use the process of telemetry. So it looks like we're carrying around one of those old cable TV antennas, tromping around the woods. When you are pointing in the right direction of the radio tag, you receive a click or a feedback. As you get closer, that becomes stronger. Now, remember I said you don't see the nests because they're hidden inside the ground or a tree cavity. But what you will see are evidence of hornets coming and going. So when we get close, that's what we look for, are the visual cues. And then what do we do once we find a nest? We remove it. That's eradication, okay? So typically eradication is going to happen late in the night or early in the morning, with the idea that hopefully most of the hornets are at the nest instead of out foraging, as well as it being, with it not being as light out there, maybe potentially a little bit more docile, because I don't want to get swarmed and stung. So we essentially seal off that one entrance they make into that nest, okay? And then we douse them with a CO2 fire extinguisher, right, to knock them out, to sedate them. Potentially it should kill them, but these guys are kind of tough. Then we attach a modified vacuum that we have into that entrance of the nest and suck all those adult hornets out. Once all those adult hornets and the queen as well are taken out, we will remove the nest by excavating or if it's in a tree cavity, by cutting that down and hauling it away. Really quick, just a couple quick pictures for you of what it looked like inside the first nest. You can see those combs there. Any one of those white marshmallow looking things has an Asian giant hornet pupae underneath it that's turned into a larvae that is now getting ready to hatch. They'll chew that silk cap off and then they will fly out and emerge, okay? Last thing I'm going to share to you what to do if you're stung. As a state agency, I got to tell you how to keep safe. So if you are stung by an Asian giant hornet, you need to apply ice, antihistamine. If you're in pain, take some aspirin and clean that sting site. If you're allergic, epinephrine and 911. It's not that serious until you're stung multiple times. So it's going to be the same thing, but seek a healthcare provider. And if you're allergic, call 911 again. They also have mandibles that can bite. They can secrete venom. And when you are stung, that skin surrounding your sting site typically does become damaged. So that is it for me. I think I am about 2 minutes close to being running out of time. So a couple more ways you can stay connected and with what's going on in the Asian giant hornet world is there's a Facebook group if you're into Facebook. Facebook.com/groups/hornets. We also have a wide array of videos on our YouTube page, as well as a recap of all of last year for you. My contact info is there at the bottom. I will also go ahead and put that in the chat box for you. But I just wanted to say thank you guys. And if there's time for questions, I'll answer them. If not, I believe there's time scheduled for them at the end.
Thank you, Casey. So it looks like you addressed all the questions in the chat box. So these are questions I will again go through because we don't have a first speaker still ready.
So there is a question in the chat box about any special PPE equipment used when we remove the nest. If you ever saw those videos or the pictures like this one you see here, of those big white looking suit things, that would be the PPE that we use. It's a couple, because their stinger's about 1/4 inch. So it's kind of some thicker foam, some really soft, thick foam, and then it's covered by like some woven nylon mesh.
Okay, so there is another question asking you is a sterile drone release program feasible?
So currently in the United States, it is illegal to harbor a live hornet. What we do though is we do take some of the hornets that we vacuum out of the nest and we do send them off for research. So the research we're doing right now with them is we have to get a special permit, to move them around. We sent them off to one of the federal labs, basically, so they could do some gene sequencing with them to see where they're coming from, as well as try to find a pheromone or a lure, something that they're super attracted to, that we could use in our traps. And then those traps are going over to Japan and they're being used in their native range to see which ones work best.
Thank you.
Oh,
So,
I totally read that question wrong. So that one queen that started the nest would be making the drones. So we would have to find the nest before being able to do that. Since the queen that begins the nest lays her own queens, and she also lays the males or the drones that come out of the same nest and then they mate together. So that would be a little bit difficult to do. We just want to get rid of them the way we are.
Have you addressed the question asking, is there any non-target species in your traps? Yes or no? I cannot hear you.
Sorry, I did answer the question.
Okay.
No, it's good.
Okay. So the lure in your trap is just the orange juice. Is that right?
It's orange juice and rice cooking wine. And the rice cooking wine is between an alcohol content of 10 to 25%.
So basically they will attract to a fermented smell, right?
I'm sorry, what was that?
So basically they will attract to a fermented smell. Okay.
Yeah.
So it looks like we still don't have a Asian giant hornet, and it is one of the lookout species here in Hawaii. So whenever it goes close to Christmas time, we have lots of Christmas trees imported in Hawaii, maybe from Washington or so. So do you have any plants there who is importing trees or the ornamental plants from Washington to Hawaii?
So most of that would come from, so we have a nurseries and a plant inspection program that does certain things like that. But as far as it going, on a boat to get to you, USDA APHIS also, looks through them and stuff. Typically when they overwinter, right? So about that time, the workers are going to be dead. So they're not going to be a problem because they can't reproduce anyway. So if a dead one was on there, that's not a big deal, as well as a male. It's going to be those overwintering queens, and they typically like to overwinter in soil. So luckily Christmas trees come over bare rooted, but if one were to get wrapped up in there and she made it successfully and made it through the winter, that could be the problem. APHIS is trained to look for them. They're the ones that do the inspections, but they made them here from Asia somehow, so gotta be ready.
So the following question is somewhat in a similar line. What do we have? It's for the Hawaii folks. This question is for the Hawaii folks, but I will just go ahead and read. What do we have in place for early warning in case these hornets appear in Hawaii? Would they be attracted to coconut rhino beetle traps? I don't know. So we have coconut rhinoceros beetle here in Oahu, and there is a area-wide trapping program. I think a coconut rhinoceros beetle has a specific lure in that trap, and I don't know if that lure will attract Asian giant hornet also.
Yeah,
if there is somebody.
I think that one's pretty specific. And also too, if the Asian giant hornet has to be actively already present around the trap, it's not gonna go and seek out that trap. It has to be flying by, because they're an apex predator, top of the food chain. They eat protein, okay? Other than those queens, when they're emerging in the spring, they like the sap, but they will stop. And that's why they are lured into the trap, because it's an energy source to keep them going while they're hunting, because they can't eat solids themselves. They have to take them back to the nest, feed them to the young, who then regurgitate it back to them.
Okay, so lots of questions. So very good. Would imported Christmas trees be potential pathway to Hawaii? If there was happen to come in Hawaii.
Yeah, so it is that fall time, right? So the queens could have potentially, if the queen had left a nest, right, and she had mated and potentially got wrapped up in a tree, and made it over there and made it through that whole process, and then was able to, come out and found a place in Hawaii that would be suitable for her to reproduce, then it could be a possibility. You know, nothing's out of the way. That also goes with like the same question of what the pathway of introduction was here. Obviously they came from Asia. So like what we're talking about, some type of freighter shipment. Also, remember when I told you that I showed you that picture, the white grubby looking things, the pupae and the larvae of the hornets. And I told you that was a delicacy. People could have brought them over for a food source and then very bad things happen. That's very illegal, but things happen.
Okay. So there's lots of questions. Yeah, we do get wasps and hornets and other pests every year in Christmas tree shipment, which typically arrive in, yes. So yeah, that's my concern also. And Dr. Cheng said probably won't be attracted to CRV traps. Okay. Xmas tree is probably low probability compared to soil. So does it comes with the soil then? What do you think?
When they overwinter, so there's only ever been 2 research papers published on Asian giant hornet, and they're both out of the 70s. Okay, this is also going to be how I'm going to answer the next question: how far can a queen fly to establish a new nest? We don't know. Oh. That's never been researched or studied. I can tell you that some species that are some of their cousins, you know, are some of the other species that are spreading can fly about 18 miles. I know that I had a queen make it all the way down to Bellingham that was dead and unmated, and that was about 20 miles, but unfortunately we have no idea. The research isn't there. The other thing too, I think I failed to mention, is that nest will produce hundreds of hornets, hundreds of queens, typically in their native range. However, it's only about 30% that successfully mate and successfully overwinter to establish a nest in their native range.
Klein, are you ready? Okay, so Dr. Klein, Michael Klein is a research entomologist at USDA ARS at Ohio Agricultural Research and Development Center at Worcester. His research primarily focused on the Scarabaeidae beetles, especially on biological control methods using entomopathogenic fungi and entomopathogenic nematodes. He started his career in 1969. That time I was never not even born. 1969, and he is retired maybe around 15 years ago, 2004. I think he's very active up until 35 years of research. And he's also adjunct associate professor in entomology at Ohio State University. And he just retired from that job 2 years ago. And today he will be speaking on the Japanese beetle that he mostly spent his career and research, not only in the United States, also in the different parts of the world, in Asia, in South America, and also in European countries. And today he will be talking about from Sapporo to Milan, a century of Japanese beetle movement. Michael Klein, all yours. Take it away. Thank you. Dr. Klein, do you have a mic?
Yes, it should be.
So yeah, you need to go maybe a little bit close to mic.
Okay.
Then we will hear clearly. Otherwise, it's a little bit soft. You have a soft voice.
Kind of, yeah.
And everybody can type in questions for Dr. Klein in the chat box as we move on.
Okay, well, this is what I looked like before I tried to log in today. But I was many years at Worcester, Ohio, and my email now is pescarabs@AOL.com. And people would come to my lab and either say, well, that's a real entomologist, or don't go in there because you might get lost and you'll never get out. So my lab is now all, and office is all closed, and I'm retired in just outside of Cleveland, Ohio. talking about the Japanese beetle, it's a native of Japan and primarily the northern part of Japan, from mainly in Hokkaido and partway down into the next province and on up into Russia and into areas like that. So it's been established there for who knows how long, and maybe probably of that area as soon as it came out. Up until recently, the Japanese beetle was noted to be in Korea and in China. But in the early 90s, a professor Ping in China found that the Japanese beetle was not present, that the species that was there was Papillia quadrigequata, and it's not the Japanese beetle. It certainly looks like the Japanese beetle, except for the one feature on the Japanese beetle, the clipius, end of the face is turned up, and it's just square across in quadrigequata. So it's hard to tell just who had the information that it was Japanese beetle, but now we know it's not. That leads me to the species Papillia. All of these are part of the some 300 species of Papillia that are in the world, and the question which one is Papillia japonica, and the short answer is none of them. But they all
What about
there's a
fingernails?
lot that look like it,
Join the meeting.
but none of those are Papillia japonica. Japanese beetle was first found in Riverton, New Jersey, just across from Philadelphia in 1916. That it was thought that it probably came in with some Japanese iris bulbs, and that's why it ended up on the East Coast instead of the West Coast. But it came in probably before 1912 when they started quarantines and looking for that type of thing. And I would say those quarantines have been fairly successful for Papillia. There's 300 species around Earth plus in the world, and this is the only one we've gotten. We must be doing something right. So the scarab philosophy is, as you go through life, take time to stop and eat the roses. And the Japanese beetle is an expert at that. It can root one beetle will bore right through the center of a rose and ruin the entire plant. And anybody that wants to discuss that, my wife had a lot of problems with telling me to go out and spray those beetles because they were ruining her roses. And I would say, go wait till the next day so we can see what they're really like. But in the northern Japan and similar cold climate, Japanese beetle has a 2-year life cycle. And that probably is true for a number of areas in the United States and up into Canada. I was in Minneapolis over the weekend, and a place that was heavily infested last fall had 1 or 2 beetles this year. So I would guess that most of them are on a 2-year life cycle. Interestingly, about 10 or 15 miles away, the trees, the linden basswood trees, were just stripped from top to bottom with Japanese beetle damage. So it's kind of hard to tell what's going on some of those areas. Maybe a 1-year cycle, mostly a 2-year cycle, maybe some overlap. So usually starting in June in the southern United States and on into August, is when you'll find the Japanese beetle adult out and about, and very visible when you see them. A lot of damage to a lot of crops, but certainly somebody said when they thought when they were coming to Ohio, they wouldn't see a single corn plant standing, but the beetles are very, very attracted to sunlight. And if you look at the outside row of some cornfields, particularly sweet corn, the beetles are quite prevalent. You go in the shade and they're not there at all. And that goes for attraction of to plants and also to the trap. So the sun is what the beetle likes until the temperatures get up in the 90s, and then they're smart enough to go underneath the leaves and feed there instead. So as I said, the flowers and my total life roses are really a favorite of the beetle. And another thing that is very common with the Japanese beetle is to see the male riding around on the top of the female. They're not mating, but they're sperm guarding. And the last sperm that's used is the first one that's used. The last one inserted is the first one that's used by the female. So a lot of males, you can see the males maybe slightly smaller than the female, and the female has a large spatula end of the tibia. And that's how you can tell males from females. So flowers are a big one most any time. A fruit is a big one, and anybody that looks closely may realize this is not Japanese beetle. And we don't grow a lot of papayas here in Ohio, but this is the emerald, Midway emerald beetle that was in Midway, and a potential one to come into Hawaii from several locations. And the foliage, this is a typical foliage. A basswood tree looked just like the ones in Minnesota this weekend, minus the blonde. Damage by larvae can be excessive, particularly on golf courses where the dead turf shows up in the fall, and usually it's skunks or raccoons that are out there digging around, but now and then the entomologists get involved too. Parwinder Grewal was working in Worcester at that time, and Kevin Powers was one of the assistants in the program. So we were out looking for beetles, and it's not uncommon to get 20 to 40 beetles per square foot. So it's something we look forward to in the fall. The superintendent, not so much. Insecticides for adults. This is a picture of what was thought to be a good idea back at the time, lead arsenic for control of Japanese beetle, and there's now a whole row of insecticides that are available for Japanese beetle control. Some can be used some places and not others. So you have to look and see. And insecticides for larvae, this is a DDT application. Both also lead arsenic was used for larvae at one time, and both of these did a very good job for a very long time, and that was part of the pleasure of using them and also the curse of using them. But until now, there are several insecticides that are used and are quite effective and safe for humans and other animals on the golf course. Quarantines are a big thing and probably something of interest there, that in the APHIS, had a national quarantine on Japanese beetle until the early 1980s, when the beetle reached Wisconsin. That tipped the balance of states that had Japanese beetles and those that didn't. And so that quarantine went away. And for a number of years, there was just kind of a haphazard use of them. But the National Plant Board then came up with the U.S. Domestic Japanese Beetle Harmonization Program. And therefore, you know, the Japanese beetle is a quality pest, not a quarantine pest, which is primarily true if you're already infested. But several people, I guess me included, also call this harmonization plan the Japanese beetle distribution program. And it's done quite well at that. Also, APHIS inspects airports, and that's a big problem for FedEx and those carriers. They've had to do a lot of work, including these covering for the opening of the airplanes and treating of airports and having people pick off beetles as they come in. At this time, everything to the east of that line is infested with Japanese beetles, pretty much in Canada, from the east too. And all of the northern states in the west have a Japanese beetle infestation. Some of them, most of them are going through a quarantine program to try and get rid of it. And these are the places that the Japanese beetle has been eradicated from. Some people say you can't eradicate the population of Japanese beetles, but all of these states have successfully done that. Some stick with California over and over again. They all around, they look like Sacramento for some reason. And of course, the big one up in Oregon, Portland, Oregon, there is a program now in Washington and also British Columbia to try and eradicate the beetle. The Integrated Pest Management Program is used to use biocontrol and exclusion, parasite traps, and insecticides. Some of a very good attractant for Japanese beetles made up of phenylpropionate eugenol geraniol, although the geraniol may be taken out because it's been shown to attract bumblebees. It didn't attract honeybees. That was tested before it was put in as a standard. But from 75 on, that's been the standard, and I don't know whether they're going to officially change or not for that. And the sex attractant for the Japanese beetle is Japanilure, this compound over on the left side. And that and the floral lure make a very good, strong attractant, not as strong as some people like to think. But if you mix the PEG and the Japanilure, for some reason, you get an increase in both sexes, in males and in females, even though the sex attractant alone does not capture many females at all. But for some reason, you get this. This is the latest research from 1981 that pointed that out. And a number of traps are available. Trace has the most sturdy trap. I think Reuters has this trap, and Bagabug, another trap. So there's a number of different traps. And some people use the trap with a garbage bag on the bottom because you can fill up a trap like this, which holds about 2,000 beetles, and you can get it to overflow in about an hour sometimes on a golf course. The traps have been used around the world for scarabs. talked about the rhinoceros beetle that is in Hawaii. And this is another beetle in far west of Japan that also has an attractant and is attracted to light green shirts, obviously. So there's a lot of good attractants out there, hopefully to use in different programs. The parasites are Typhia wasp, the spring Typhia in particular, Typhia vernalis. And the most important biocontrol in Japan is the Tachinid fly, Istakita aldrichi. And that fly is now being moved around the United States by Richard McDonald in North Carolina, particularly, and is establishing in a number of locations. Diseases, bacteria, milky disease, Penicillin pili has been around for a long time. Amber disease is one that's found primarily in New Zealand on the grass grub, but you do see it now and then in the United States. Nematodes, two different genera, Heterodytis and Steinerdema. The color is caused by the bacterium that they carry. And this was actually a new species that was found on a golf course in Ohio, Heterodytis megatus. So you've got nematodes, you've got bacteria, you've got fungi, Metarhizium anisopri and Bavaria bassiana. If you put them in a trap, particularly the Metarhizium, either with 1 or 10 milligrams of material, that you get a very, very good and quick death of the beetles that takes them out of the picture. And that is an auto-dissemination process by which the adults also move the pathogen into the larval environment. We use a trap like this where the beetles come in here. They're very, as I said, very attracted to light. And so this is a screen at this end and the petri dish with the fungi in here. And as they walk towards the light, they fall out and disperse. Talk a little bit about the Azores. It's an interesting place, an interesting island, a beautiful place to be. But as the Columbus poem goes, behind him lay the gray Azores, or Azores. And if the Beatles read the literature, they would have known that they couldn't establish there because temperature never gets high enough for them to fly. But somehow they've adapted to that and did quite well. They were found originally on an air base on the eastern end of Terceira Island. It was an air base for the U.S. military. And although they claimed they did not have any part in bringing them there, out of the goodness of their heart, they gave the Azorians $1 million to deal with it. One thing that happened was that they have everything's a pasture with stone walls around it and with blackberries and other food sources on it. And lots of rain, very green, a great place for Japanese beetles. So what could possibly go wrong? Well, the general's wife, they wanted to turn off the water on the base and dry out all the lawn. And she said, if I have to be on this tiny rock, it's going to be with a green grass. The generalissimo said, you can't treat the air base because I run my cattle out there. APHA's advice told them that don't worry, the beetle won't leave the base and go up into the lush green environment before it. Department of Defense, I mentioned, was not too happy with the whole situation. Beetles leaving the island, the guy in charge of quarantine and movement of beetles, prevented beetles from going off the island, was also the only nursery on the island that moved things off the island. So beetles tended to go that way. And interagency cooperation between state and universities sometimes was a little strained. And a hurricane came through just as we were about to do a big program on one of the islands. So that's those are some of the little things that can go wrong. So with $1 million in the infestation here in the early 80s on Tercero, we took that $1 million and managed to spread the beetles to all the islands by 1910 with the, except for one down here. The beetle has never moved from these islands back to Portugal, but it does create a problem and still is a problem in most of these areas. So from the Azores, the beetle showed up in just outside of Milan, Italy, sorry. Milan, and when they put out traps, they caught between 8 and 10 million beetles. And you would have thought that as visible as a Japanese beetle is, that maybe someone would have seen one before that time and managed to look at something. But 8 to 10 million beetles, and there's word that the beetle has spread to some of the islands in southern Italy. And this summer it was reported that the beetle had jumped from the bottom of Switzerland over the mountains and up to Basel on the border with Germany. So I think that we can expect the other areas in Europe to experience some real problems in the next few years. So thanks for the invitation. A little limerick of the Japanese beetle. As I said, the people in Japan are very fond of the beetle. They think it must be a very rich beetle to afford such beautiful clothing. The people in the United States, there aren't enough cuss words to cover the ground. So if the insect that's on the move, I wouldn't say no, but I doubt that it would do that well in Hawaii. It's down as far as Florida on the east coast, and I think it's entirely a convenience that Florida hasn't looked for it, so it hasn't been found, so that they can ship things more freely from there without the quarantine and the problem. Because once the dieldrin and chlordane and those materials were no longer available, there's really nothing that gives 100% control of nursery stock and other things that are moved. So it's been an interesting voyage for me, and one that I kind of sit back and look at and enjoy the view for now. So thanks for your attention.
Thank you, Dr. Klein. So we have lots of time for question answer. So before we go to question answer, I have one more thing, one more quiz, one more poll. This will be the final one, I promise. Here I will launch and it will stay for 1 minute. It has around 3 questions. So Dr. Klein,
Yeah,
so trapping is the one best way to control Japanese beetle, yeah?
but trapping is, I would say, the best way to find Japanese beetles. There are problems with beetles being attracted to the trap, but not going in the traps. It's not as bad as some people would think, but it is a concern, and people, when you have to keep the traps away from preferred hosts, or they'll start feeding on the host, which then makes it very attractive. But for the quarantine work, California uses 10,000 Japanese beetle traps a year, 2 per square mile. And that's kind of the standard for the West to use to find a population, to delineate it, and then to move in. But most people will spray for adults, not nearly as much treatment for larvae.
So my second part is, so trapping is very effective and it's one way to control. So I think I can see when the Japanese beetle goes to trap, there are lots of beetle goes to trap, yeah? Is there any spillover effect? So you put the trap, you attract beetles, and beetles will go somewhere else. You will invite beetles to infest other crops. Is that the case?
Particularly if the trap is near a favored food host, or if it fills up and overflows, then the beetles go somewhere else. And one problem with the bag traps is that it doesn't take very long before the beetles rot, and that repels beetles. But yeah, you can. Some people, like traps. It may be more of a revenge than control, but every beetle they see in a trap is not on their bushes, but they can attract beetles, but probably 100 yards or so. If you put them on a golf course, you can see beetles kind of rising up 50 or 100 yards down. But you've got to somewhere where you've got to have a continuous plume like that. So probably not as far in neighborhoods.
Okay. So yeah, questions for Dr. Klein and Cassie as well. So yeah, we have lots of time left for questions. If anyone is interested to ask question in person, you can unmute yourself and ask also.
I have a question.
Mhm.
Do you know why, does anyone know why the Japanese beetle harmonization plan uses like 3 miles as a parameter for establishing like from a detection? Because I was trying to find like the maximum flight distance for Japanese beetles, and all I could ever find was fishermen at sea being swarmed about like 5 miles off of shore. So I was wondering why the harmonization plan uses 3, and if anybody knows any way how to answer my question.
Yeah, beetles. That 5 miles, they said beetles have been found offshore, and the trapping in Tennessee with marked beetles, one was found 5 miles away the next day. But that doesn't mean that it was attracted for 5 miles. They can fly about 5 miles. The zone of infestation tends to move about 5 or 10 miles a year, but from the often used traps in California, they came up with that 2 traps per square mile, and then changed. If 2 beetles were found, they went to 50 traps, and then 25 and 5 in the areas around it, as you say, the 3 mile area. And that seems to have been really effective. There have been very few flyouts of beetles to those outer edges. It does happen, and then they have to restart from there, but very seldom do they fly out with 50 traps in the middle. And we've released beetles on golf courses, and you can find them, 500 or 600 miles away, but in not miles, 500 or 600 yards away down a fairway the next day or something, but they don't fly out unless they're something might agitate them and they would fly, or they would go on the wind, but they usually fly into the wind to an odor, so that's a little different. But I hear you have an infestation in Washington.
Okay, great. There is some more questions on traps. Are traps best for all species of beetles in general?
A lot of the species, a lot of scarabs have had pheromones identified, and some with, like the Japanese beetle, with what are called the floral lures, so that combination. But a lot of the scarabs work was done in Japan by Walter Leal, that identifying the attractants for many of the scarabs that are found in Japan.
So this is pheromone, not lure, yeah, for the beetles.
What's a combination for the Japanese?
Okay. So for the Japanese beetle, there is three chemicals involving.
There's three,
Eugenol,
yeah,
something. Okay.
yeah,
One of them is pheromone.
yeah, and the pheromone.
Eugenol is pheromones.
Eugenol,
Mhm.
phenethylpropionate, eugenol, geraniol, and the pheromone. But
So, okay.
the geraniol attracts bumblebees and is not a good component. It really didn't increase the captures that much.
There is another question about trap. Are the traps effective at detecting beetles at the port? So in connection to this, I have another question. You showed us a beetle moving from island to island, yeah?
Yeah.
In Portugal. How does how that can be possible, or how they move from island to island, including traps at the port?
Yeah, I think that some of the islands are close enough that the beetles can fly, that they would get carried in the wind or in things like that. Others, the quarantine efforts are not very good, and there's a lot of movement of hay and things like that from one island to the other. And although that isn't really a particularly host for beetles, they certainly have been moved that way. And one of the first islands that was infested, their second island, has a lot of yachts that stop there and go different places. And they probably brought it in, but was found, in the yacht harbor. And then that's the one where we were all set to do a program, and then a hurricane came through and all the money went to rebuilding the island, and now the whole island is pretty much infested. But yeah, traps can be placed at ports. They would pick up things that come. I don't know how well a beetle would survive, on a boat or something like that. Easily there are boats that go between the islands there, so it would move inter-island pretty well, but not so much from the United States or from Asia to come into. I think that our friends in the Defense Department, I would watch those airports.
Okay, thank you.
They haven't figured out exactly where the beetle came from in Italy. They've done a lot of studies, and it looks like it did come from the United States and not from the
So again, one more, just curious. If the beetles move from one place to another, island to island, or country to country, is that at the egg stage of the insect in the soil?
Either the larvae or the adults could be moved.
Okay.
Probably more of the larvae. It's interesting that scarab beetles have not moved from the northern hemisphere to the southern hemisphere, or vice versa. That If a larvae is in the soil, it gets to New Zealand or something, it's the winter and they can't continue or survive. So that's one thing. Now the Erichthys beetle is more, I guess, in the Hawaii atmosphere. So the question about JB and soil and yard waste, and yes, that is a way that they're moved. And in the quarantine programs, that's always part of it, that all yard waste is picked up and disposed of rather than just putting it into a landfill or something like that. And that's the main way that they've moved in the soil with the plant protection harmonization agreement is with nursery stock. It moved into Minnesota, and then a nursery there distributed them nicely to South Dakota and North Dakota. So that kind of thing happened.
So Dr. Klein, you have worked in some South American countries also, yeah, where there is a tropical climate. So what is the difference in the life cycle of Japanese beetles in tropical places than the colder area like Canada in the United States?
In the cold places, it's a 2-year life cycle. There hasn't been a place that the beetles have been where there's a 1-year life cycle. I mean, less than a 1 year, a multiple life cycle, so that even when the beetles were in San Diego and now down on the border of Florida, there's just one generation a year, not more. So there was a question about fungi, I think, and that's one of the, I would say at this point, one of the bigger biological control agents. There are several people that produce fungi for both adults, but particularly for larvae. So Metarhizium is one that's been used in a number of places by people that want to use biological agents. As I say, the fly is really quite good. It parasitizes females in preference to males, probably because the females are used to something riding around on their back anyway. But flies come in and lay an egg, and it's 3 to 5 days that the beetle dies. So if you go weekly, those are all new beetles that are being infested.
Thank you, Dr. Klein, and thank you for this question. I think this is the last question, yeah. So going back to Japanese, Asian giant hornet. I think I'm checking. So going back to Asian giant hornet, our Chris Kishimoto wrote something. So far, the areas in Washington where the hornets are found are not close to where the palms that supply Hawaii's tree come from WSDA, Washington State, come from, and WSDA also inspects trees shipped to Hawaii, and SDOA also inspect those trees when they arrive here. Thank you. And there might be another question for Cassie. Is there any commercial? So do you know?
Answer that question in the private chat.
Okay.
Yeah, there's no commercial trap.
Just orange juice. Yeah.
Can you hear me?
Just orange juice, yeah.
Yeah. So it's based out of Japan. No commercial traps, unfortunately. That lure is still being researched. So we aren't quite yet as cool as JB.
So is there any sensitive? You said there is 3 detection all removed. We had 3 detection and removed 1 nest last week. So now you don't have Asian giant hornet in Washington?
Well, I mean,
it's a silly question. Yeah.
Yeah, hopefully we don't. Unfortunately, if we missed any nests last year or any queens got out before we removed the nest from last year, when we did open that nest, we did find queens in it. So if they got out and started new nests, they could be out there. So we're still asking people to trap and report for sure.
Okay. Okay, thank you. So orange juice and rice one lure for the, okay, yes. So one more question for Dr. Klein. I saw a taconite fly. Is that taconite fly lay egg on the Japanese beetle, adult Japanese beetle body?
It lays the
Egg.
lays on the thorax of the female. So it's a white egg on that green thorax. It shows up really nicely when you go to collect them. You're collecting them and moving them to places in the West.
Okay, thank you. Yeah. So they hatch and they just feed on the external body and go inside. Is that the way they kill them?
Drill through the thorax, which is pretty hard, and then they feed internally, and the beetle dies within about 5 days. So I said, what's the most prominent biocontrol in Japan, and one that we're trying to use more in parts of the United States.
Okay, very good. Thank you. Thank you, Dr. Klein, and thank you, Cassie, for your nice presentation, and you have a good discussion too. Thank you. So we just move on, and we have another speaker, another presentation from Mark Hughes, laurel wilt disease, a potential threat to avocado trees in Hawaii. Mark, are you around?
Hello. Yes, I'm around.
Yes.
I'm just going to turn my video on.
Yes, Mark Klein is our, not sorry.
Oh, go ahead.
Mark Hughes. Mark Hughes is our, I think, only lone forest pathologist here in Hawaii. And he's with USDA Forest Service, Institute of Pacific Island Forestry. And he is also affiliated with Pacific Cooperative Studies, University of Hawaii. And basically, his current research is focused on rod management in Hawaii. And he also has lots of experience in laurel wilt disease before this job. Okay, so he will be talking about laurel wilt disease. This is not here in Hawaii yet, but we still need to know, and we make ourselves ready when it comes to Hawaii. Thank you. So, Mark, please go ahead.
Great, thank you. Can you see my screen okay? Okay, good. Okay, so yeah, today I'm going to be talking about laurel wilt disease, and it is a potential threat to the avocado industry here in Hawaii. So I'll just start with a really quick introduction of myself and my experience of laurel wilt disease, and then really just get into the biology of laurel wilt, talking about how it works in the forest setting, and then also how it works in the avocado groves. It's kind of a different mode of spread, and then talk about some potential management options, what's being done in the avocado setting in the avocado groves of Florida. So we can go through those potential options. So just a little bit about myself. I started my PhD work at the University of Florida in 2007. And I worked on laurel wilt disease in red bay. So red bay is the predominant forest host of the disease. It is, so it's the one most affected. And sometimes I'll say red bay and swamp bay interchangeably. They're kind of sister species, but they both have same levels of susceptibility. So my work looked at evaluation of natural resistance. So is there resistance to this disease naturally occurring in populations of red bay? But then beyond that, just through lots of collaboration with researchers in University of Florida and the Southeast region, we looked at management strategies, chemical ecology of the vector, and fungal population genetics. And okay, I'm sorry, I'm looking at the chat screen too. So let's see, can we? Okay, there we go. So just kind of any plant pathology talk, you got to give the talk about the disease triangle. This is a graphic that illustrates what you need for what conditions need to come together for plant disease to occur. in general in nature, disease in plants is a rare phenomenon, but if you do have a susceptible host of favorable environmental conditions and the pathogen all coming together, and with this case in laurel, you need a vector carrying the pathogen. If it all comes together and those conditions are right, you're going to get plant disease. So for laurel wilt, your susceptible host is members of the plant family Lauraceae. So that's the laurel family. Again, I talked about red bay and swamp bay. Along the southeastern United States, there's a lot of Lauraceae, but here in Hawaii, the major host, the one of concern, would be avocado. Favorable environment, as of right now, we don't really, it seems like the range of Lauraceae is fair game for laurel wilt. We don't really know. In the mainland, the disease hasn't really expanded into areas cold enough to test the upper limit or the lower limits of temperature. It's still moving along in that direction, but we don't know. So as of right now, I'd say it's fair game. Hawaii is definitely fits within that temperature range that would be favorable for laurel wilt. And then you have the vector and the pathogen. So the primary vector is the red bay ambrosia beetle, which is Xyloborus glabratus. There are some other ambrosia beetle species involved, especially in the avocado side, which I'll talk about. And then the fungus, the fungal pathogen, is Rafflesia loricola, which is an ambrosia beetle symbiont, and I'll talk about that shortly. But it's the predominant fungal species associated with the red bay ambrosia beetle. So I'm mentioning it's a dual-faceted disease because the vector and the way it's spread is very different in the two systems. In the forest tree setting, which I did my work predominantly focused on, the hosts are red bay and swamp bay. So these are Persea species. And then sassafras, which is another member of the Lauraceae, and it has a much larger host range. And that's kind of where the disease is expanding into new territories outside of the range of red bay and swamp bay along the East Coast into the range of sassafras, which is much larger. It goes even into Canada, I believe. So we don't know how far it can go. In the forest side, there's about a dozen species of Lauraceae that have been shown to be susceptible to the disease, either through natural infection or through artificial inoculation studies. And the method of spread in forest trees is predominantly through movement of the vector, the red bay ambrosia beetle, but there is evidence of root graft transmission in sassafras. And then there's also wood movement, which could potentially move the vector around, causing new disease hotspots. With avocado, it's a little different. Persea americana is the host, and avocado is the plant itself is composed of it has three different genetic backgrounds. It's West Indian, Guatemalan, and Mexican backgrounds, and then there's different cultivars or different hybrids of all of those genetic lineages. I would say that the main and most problematic method of spread is this root graft transmission, which I'll talk about, and that's predominantly within an avocado grove once the disease has gotten established. And then there's this secondary spread by ambrosia beetles. So when we talk about laurel wilt in forest hosts, I'll kind of start from this distribution map, starting from the beginning of laurel wilt being known in the world, basically. The red bay ambrosia beetle, Xyloborus glabratus, was first detected in 2002 in some monitoring traps at Port Wentworth, which is right near Savannah, Georgia. And there was already this kind of mysterious mortality occurring of red bay and swamp bay trees in the area, but they thought maybe it was a severe drought or something like that. But as the entomologists and the pathologists got together and investigated further, they found that this beetle was always associated with this fungus, Rafflesia loricola, and that fungus can cause disease. So they put the pieces together and figuring out that this is a new disease system called laurel wilt. And fairly quickly, the pathogen and the mortality caused by the beetle and fungus spread throughout the East Coast. And all of these counties are pretty much now infected, anything that you see with a swatch or a color. And just to highlight, wood movement is a potential method of spreading the pathogen and vector. As you can see here in 2006, there's this large jump from kind of northern Florida. And that is a case where there was somebody was traveling through the area and they found a wood pile, infected wood on the side of the road. They looked at it, they saw it had this beautiful black staining in it, and they said, I'll take this wood home and I will do some wood turning and make some bowls out of it. And so I'll just throw it in the back of my truck, take it home, put it out in their shed. And, you know, beetles popped out and started lower wilt disease down here. So it's a good reason not to move firewood around, especially, you know, unprocessed wood. The lower wilt disease cycle and the disease or the vector life cycle are fairly closely tied. What you'll have is this ambrosia beetle, the red ambrosia beetle here. It's carrying the fungus already in it. It's mycangial symbiont, which I'll talk about shortly. And it's attracted to healthy trees. It will bore in. During that boring process, it inoculates the tree with the fungus. It's a very virulent vascular wilt pathogen, so it's going to quickly spread throughout the vascular system of the tree. And then once the tree dies or is on its way to dying, it's very attractive to more red ambrosia beetles and other ambrosia beetle species. So they get, you know, attacked more. The beetles make their gallery system, so they bore tunnels all throughout the tree. They lay the eggs. The fungal symbiont of Rafflesia loricola is all throughout these galleries, and the larvae feed and develop on that fungus. And then they mature, they fly off, and they infect new trees. And that is the disease cycle. So for those of you that aren't familiar with an ambrosia beetle, it's essentially a tiny wood-boring flying beetle. So only the females fly. But what's kind of unique about them is they carry and rely on for their life these fungal symbionts. So the fungi they carry is what essentially feeds their larvae when they make these new galleries. They're not like bark beetles that can feed on phloem tissue and for nutrition, even though bark beetles do carry fungi too. These are specifically reliant on them for their life cycle. And sometimes, like in the case of the red ambrosia beetle, that symbiont happens to be a very aggressive pathogen. In general, these beetles are attracted to stressed or dying trees. Again, oddly, with the red ambrosia beetle, it seems to be attacking healthy trees, but again, they're generally attracted to stressed and dying trees. And some can be very host specific, and others are just generalists. They can attack all sorts of families of stressed and dying trees. So lower wilt and swamp A has, if anybody's familiar with Rapidohea death, that's another disease system. Well, that's the main disease system I work here on in Hawaii. It's similar to Rapidohea death caused by Ceratocystis leucohea. So it's a systemic vascular wilt disease that if you were to open up the sapwood, you'd see this extensive vascular staining, which is an indication of fungal colonization. You're going to get wilt in specific segments of the crown that will progress throughout the tree very quickly. And then you're going to have trees that are wilted and dead with the leaves generally staying on for about a year. So in total, lower wilt in the forest setting has been very devastating. We did a study looking at forest inventory data of the abundance of red bay before and after lower wilt, and it was estimated about 300 million trees have died due to lower wilt disease in just that maybe 15-year span since introduction or first detection of the beetle. So this really highlights that these invasive pathogens and pests, you might not even know that, oh, this is a species of concern. These combinations that you don't really think of can occur. And lower wilt doesn't cause much disease in Asia. It was only recently found to cause disease in avocado in Asia around 2017, but there's no lower wilt occurring in native forests where the beetle and pathogen are from. So it's only when you bring these organisms to new areas, have them interact with new hosts, that sometimes you get these terrible combinations of disease that occur, which is also, again, something very similar with Rapidohea death that's coming here. So again, it shows that devastating potential of exotic organisms. So now kind of shifting gears to avocado, I want to let it be known that only in Miami-Dade County in the grove setting does lower wilt exist. There are people with residential trees along, the state of Florida that have lost their trees of laurel wilt, but when you're talking about production groves, it's only in Miami-Dade County down here, the last county. It's not found in Mexico, which is the world's largest producer, or California, which is the United States' largest producer. The disease nor the vector are found there. Moving through the timeline, the vector was detected in 2002. By 2010, it has moved far south that it's about 21 miles outside of the groves. A year later, the first red bay tree was detected to have laurel wilt in the vicinity of the groves, and a year later, laurel wilt actually started to attack trees in avocado groves. So luckily there was a good amount of time for the growers and the scientists that worked on avocado to start preparing strategies and learn about the disease. And I think it was very beneficial for these researchers and the avocado growers themselves. So leading with that is that a lot of the research that's been done with mitigation management of laurel wilt in avocado groves has been done by people at the University of Florida. They have a tropical research station down in Homestead, right in the neighborhood of where a lot of these tropical fruit trees are grown. So I'm going to disseminate some of the knowledge that they've learned over time. And this is also an extension publication. If you want to do further reading, this is a really good, concise document for that. So as of right now, there are 32 cultivars that have succumbed to laurel wilt, mature trees in the groves under natural inoculation settings. In South Florida, the majority of the trees grown are of West Indian or West Indian Guatemalan background. Looking at some information from the Hawaii Avocado Association's cultivars most popularly grown, you can see that there's no real overlap. It seems like what's grown in Hawaii is not grown in Florida, which leads to a question of, first, what is the genetic background of these cultivars? And it's unknown how susceptible they are, because even though most cultivars we think are susceptible to varying degrees, the question is how susceptible, So that's another avenue of research that can potentially be followed up upon. Symptoms of laurel and avocado. You have a similar wilting of green foliage that progresses to that brown crown, like we saw with red bay. You can see, if you look closely at the bark, and lots of evidence of ambrosia beetle attack, which are these, what we call frass tubes. So this is the sawdust that is being ejected when the beetles bore into the tree. And you get this vascular staining also, similar to many vascular wilt diseases. What's different with avocado that doesn't occur in red bay is you can get a lot of leaf drop occurring once the tree is infected, and which can much faster lead to defoliated trees. With the forest host of red bay and swamp bay, you generally have the trees keeping the leaves on much longer. Okay, there we go. So I'm going to get into some data now, and I'm going to try to walk you through it. So just bear with me. One of the first things that they realize is now there's laurel wilt in the groves, and it's attacking avocado trees, and it's attacking swamp bay trees. So the entomologists, and this is particularly the research of Daniel Carrillo from University of Florida. I've seen him here in Hawaii, so he does do some collaborative research, not on laurel wilt, but I believe fruit flies. So it's a good community, I guess, lots of intermingling. So the first question was, how are these ambrosia beetles interacting with these two hosts? What is the abundance? What is their preference? So what they did is they took ambrosia, laurel wilt affected avocado trees and swamp bay trees. They cut them down, and they cut them into pieces of wood that they call bolts. So it's just a section of wood. Put them in these rearing chambers, which are just plastic tubs essentially. And they collect all the beetles that propagate and emerge from these pieces of wood. And that gives you an indication of what beetles are using this wood for reproduction and brood development. And the first thing that was really odd and surprising to everyone was that the red bay ambrosia beetle, the main host in the forest, is not being captured in the groves in avocado here in the orchards, and it's not being reared out of avocado trees. If you look in the same area, geographic area in Swamp Bay, you could see that, out of 53,000 beetles that were reared out, half of them are coming from the red bay ambrosia beetle. The story kind of evolves in that there's all these other generalist ambrosia beetle species, which are also attacking the red bay ambrosia beetle, Swamp Bay, which is, again, they're generalists, so it's not very surprising, but they seem to be causing a lot of damage also in avocado trees. So I think the first twist in the story that was unexpected was that the main vector in the forest setting is not seeming to be too much attracted to avocado trees, especially in the groves. So that was, again, one of the twists. And taking the next step forward, you think, well, okay, so these beetles are emerging from Swamp Bay trees that are infested with the laurelwort pathogen. But the question is, when they emerge, when they come out of these pieces of wood, are they actually carrying the pathogen with them? So this table is looking at recovery of the laurelwort pathogen from the beetles that are reared from diseased Swamp Bay trees. So if you look at the percentage of beetles with the pathogen, again, the red bay ambrosia beetle, the main vector in the forest, it's about 86%, which is fairly obvious because it's the main vector of and that's its primary fungal symbiont. If you look at the CFU, which is the colony forming units, okay, sorry, just looking at the chats. It's not obvious on my screen. If you look at the colony forming units, you can see that it's anywhere from 2,800 to up to 7,800 spores obtained from the beetles. And even in earlier work, it's shown that they can carry even up to 30,000 spores. So they carry a large fungal load. But these other beetles, these generalist beetles that are also attacking Swamp Bay trees, you're still getting some of them acquiring the fungal pathogen. Even 50% or more of these individual beetles are acquiring the pathogen. But the numbers, if you look at how many spores they're leaving the tree with, is much lower. So again, it's just more of an inefficient vector, but it's showing this case of a lateral transfer of the fungus from one beetle species to another. And what's happening is, if you remember, the trees here at this stage, when they're dead and dying, they're very attractive to the red bay ambrosia beetle, but also all these other beetles here that are hanging out in the area, they're also attacking the tree. They're also making galleries and living in the tree that is thoroughly colonized by the laurelwort fungus. And then they're also flying out. So that's where that transfer is occurring, we think, is in these host Swamp Bay trees. Excuse, there's a little bit of a delay. So looking at the same thing of these beetles that are leaving trees, how much of the pathogen are they leaving with? So we're looking at avocado trees, and the story again shows that the red bay ambrosia beetle, Xyloborus glabratus, is not being found in avocado trees at all. It's not being reared out of these laurelwort affected avocado bolts. But if you look at particularly Xyloborus bispinatus, Xyloborus ferrugineus, Xyloborus volvulus, you're getting anywhere from 19 to 100% of the beetles are having detectable fungus on them, albeit at very low rates comparatively. So the next question would be, well, we know that these other beetles are leaving or have the ability to leave laurelwort affected avocado trees, but can they actually transmit the fungus? So again, Daniel Carrillo and his lab did an artificial study where they reared out beetles from laurelwort affected red bay trees and essentially forced them to bore into containerized avocado and red bay seedlings. And they looked at two things: transmission, so can they detect the fungus in the tissue of the plant, and laurelwort development. And what is pretty obvious here is that all these other Xyloborus glabratus and all these other species can infect red bay trees and cause laurelwort. And again, that is really a testament of how susceptible red bay is as a host. Xyloborus glabratus can also infect avocado tree if forced to bore into it, probably a function of its high spore load that it carries relative to the others. But what was really surprising is these two other species, Xyloborus volvulus and bispinatus, are also able to transmit the pathogen. And that's leading now to this thought that these are potentially secondary vectors in the system. So the working hypothesis of how this occurs is that you have in avocado groves of how do these groves get infected is, well, you might have native swamp bay and red bay in forests that are spatially nearby avocado groves. And you have the red bay ambrosia beetle attack these trees, put in the laurel wilt fungus, and then all these other generalist species come in and they all inhabit the infected swamp bays together. That's when these other ambrosia beetles acquire the fungus, and then they're going to go, and these other beetles are much more attracted to avocado. So they're going to go in and they're going to attack avocado. A lot of times avocado gets stressed because of flooding events, Phytophthora root rot, lightning strikes. All of these are things that can cause ambrosia beetle attack. And there's even some evidence that none of that's even needed for minor attacking to occur. So these secondary vectors come in, attack the avocado trees, and then you're going to have transmission within the grove in two different methods, either beetle treated tree attack. If your grove is not managed and you're not trying to eliminate these beetle populations quickly, a standing tree is a beetle factory. So you're going to get transmission and propagation of these populations. So you could get spread that way. And then the other spread is root graft transmission. In Florida, the groves are planted in rows. The trees are very old, decades old. So there's this fusing of the roots, and the fungus can move through these root systems like that. Root graft transmission, again, this is showing some photos of how it occurs. It's very common for vascular wilt fungi to move through these root grafts. It happens in oak wilt disease, Dutch elm disease, even laurel wilt and sassafras. And it could be very problematic in avocados because, again, of the way they're laid out, their grid system, it's been shown that you can get about movement of the pathogen of about 3 to 6 trees per month, if not dealt with. And in a year, you can get about 90 or 6 months, you can get 90 plus trees infected. And they see this a lot in certain groves that are just abandoned. If you don't manage for laurel wilt, the grove, you could just see laurel wilt occurring right down the row, tree after tree after tree. So it's a very efficient way for the pathogen to spread. So looking at suggested management options in Florida, and this is what they've developed in Florida, and how does this translate to Hawaii? That's a great question, but I think it's all good that we're all knowledgeable of what's being done, because compared to what's occurring in the forests in the mainland United States, laurel wilt in the avocado groves is not nearly as devastating. It's devastating if it's in your grove and you don't do anything about it, but it seems like the management strategies can work if implemented in a timely manner. So one is just to promote healthy trees. If your trees are more healthy, they're less likely to get attacked by ambrosia beetles. Scouting is done. They can do that with drones or just by the person looking at the canopy. And that's really, you want to minimize the time for ambrosia beetles to produce brood and multiply, and also minimize the time for that root graft transmission to occur. One of the main ways is to rogue the tree. Is essentially you have to uproot and chip up the entire tree. And I know, talking to a grower in Hawaii that has very large trees, that could be a very difficult task, but that's what they do here in Florida. And really the main reason is because it disrupts the root grafts. And again, if you don't disrupt these root grafts, you can have movement of the pathogen through them. And it also reduces ambrosia beetle populations. So that's one of their main methods for treating infected trees in a grove setting. You can also use prophylactic fungicides, not on your tree that is infected, but if you have trees down the road that are in line from getting transmission via root grafts or spatially near trees that are infested with ambrosia beetles, you can use these fungal and fungicidal injections, which will protect the tree. lasts about maybe 24 months or 12 to 24 months. You can do insecticidal sprays. Remember, if you chip and shred wood, that's a great lure. So it's going to bring in lots of beetles. And if you spray them, you can kind of tamp down that population. You can also spray these insecticides on the trunk of areas. If you have laurel wilt already in your grove, you can do this to healthy trees nearby. Other methods that they suggest in the Florida setting is to prune the trees to maximize sunlight to the lower, to the floor of the grove, because they found that the more sunlight, the more it suppresses ambrosia beetle population. So if there's ways you can prune these larger trees to increase the sunlight penetration, that could be helpful. They also suggest maybe rejuvenating these older, very tall trees. And this is, again, kind of drastic, and I don't know how feasible it is in Hawaii, but you can stump it, which essentially cut it back to 4 feet and then let it regrow, or hat rack it, which is, again, cutting it back to about 8 feet. And I guess one of the upsides, other than letting the exposure to light increase, is that you can re-top graft onto these new stumps. So maybe if you were growing a cultivar that wasn't as profitable or desirable, this could be an opportunity to maybe plant like Charwell or one of those other cultivars. It's going to take your trees out of fruit production for about 3 years, at least in the Florida setting. I'm not sure how that would be in Hawaii. And if you do have trees that die due to laurel wilt, you can replant them. At this point, they're too small to be attacked by ambrosia beetles. They're not going to have root grafting contact, and you're going to have that high light exposure. So again, I've had a conversation with a local grower, and just talking about the situation of laurel wilt here in Florida. You could see there are these blocks in Miami-Dade County of just huge acreages. So there's a lot of resources that can be put behind managing this disease. In Hawaii, it doesn't seem like that's the case. It seems to be much smaller scale farming, a couple acres maybe per grove. So again, you might have limited resources by the growers themselves to enact these more substantial managements, like cutting the tree down, chipping it, uprooting it, especially in regards to tree stature. This grower is mentioning that all our trees are pretty massive. So again, how is that going to be accomplished? I think that's something that needs to be thought out and considered moving forward, again, with this feasibility of rotating trees. And I only have a few seconds left, but I was trying to think of maybe some encouraging thoughts, and really maybe what are some natural barriers occurring that why hasn't laurel wilt come to Hawaii yet? And I think the whole meeting, the idea is that these insects, there's always a chance they could come here. So I'm not trying to say that laurel wilt cannot come here. Knock on wood, I hope it never does. But some of the natural barriers that are maybe helping keep laurel wilt away is one, that it's a really remote island chain. So it hasn't, the beetle or the pathogen hasn't infected California or Mexico yet. And it's much closer than Hawaii. So there's a good spatial barrier here. The beetle cannot fly across the ocean. They only fly generally a few meters, maybe a mile. So across the ocean, it's not going to happen. Other avenues, and when you think of transmission of diseases, is more maybe symptomatic plant material. Looking at the cultivars, comparing what's grown in Florida and Hawaii, I don't see much overlap. So I really don't know what the growers, how they're sourcing the material, but it seems just based off cultivars that they're not getting the material from Florida. And much less seedlings are not really commonly attacked by the red bay ambrosia beetle or these others. So I don't see movement of plant material as being the highest risk, but again, anything can happen. So I'm not saying that these are non-issues. They're definitely issues, but I'm just thinking out loud of what are these potential barriers. Another is limited Lauraceae in Hawaii. It seems like Hawaii just does not have this natural reservoir of hosts that can hold large populations of the red bay ambrosia beetle. There's a vine, which is not going to be a good host, a rare forest tree species, and then cinnamomum, which is There is Cinnamomum camphora in Hawaii, which is camphor tree, and that is kind of a bad host in the mainland. So again, it's good that Hawaii's not this luscious Laraceae, abundant place, because it means that it's less likely to have these alternative reservoir hosts. And with that, I'd like to thank you for your time. If you have any questions, feel free to email me. My address is right here. There's some good resources here in regards to the University of Florida, what work they've done with Laura Wilt in the avocado systems. And from that, I'm 3 minutes over time, so thank you. And feel free for questions.
Thank you, Mark. We will do question and answer session after another talk, which will be last talk, and we will have some like 20 minutes left at the end. So the next talk is on two-line spittlebug. So this talk is second time in this mini conference. That means something is going wrong in the Kona area, Big Island. And today we have Shannon Wilson talking biology and pest status of two-line spittlebug in Kona. So Shannon Wilson is a graduate student, now PhD in Department of PEPS, Plant Environmental Protection Sciences, with Dr. Mark Wright, and she is also working at the Kona with Mark Thorne as well. So Shannon.
Hi, let me share my screen.
Okay, Shannon Wilson here. Whenever you are ready.
All right, can everyone see that?
Not yet. Okay, now
Not
yes.
yet.
Now yes, yeah.
Good. Okay.
No. Can you do full screen?
All right. What's that?
Slideshow. Oh yes, now yeah.
Okay. So today I'll be talking about the biology pest status of the two-line spittlebug in Kona. I'd like to begin by thanking Dr. Mark Wright and Dr. Mark Thorne, who have led this research project. Spittlebugs are hermapteran plant-sucking insects in the superfamily Cercopoidea, which are considered the most injurious and widespread economic pests of forage grasses in the New World tropics and subtropics. The two-line spittlebug is in an economically important family and genus. Several species within the Cercopid family are responsible for inflicting significant economic damage to pastures and sugarcane throughout Central and South America.
Hey, Shannon.
Thus, grassland cercopids are regarded.
Shannon, your voice is breaking. Are you away from mic, or do you have some other?
I think it might be the internet connection.
Okay. Go ahead. Thank you.
Sorry. Several species within the Cercopid family are responsible for inflicting significant economic damage to pastures and sugarcane throughout Central and South America. Thus, grassland cercopids are regarded as the most destructive pasture pests and the second most destructive sugarcane pests. In the economically important genus Prosopia, 3 of its 15 species are of substantial concern as forage pests. There are no native spittlebugs in Hawaii, and while the two-line spittlebug is the first cercopid to invade the state, it is not the first introduced spittlebug. There are 2 species present in different families. However, neither has inflicted economic damage comparable to the two-line spittlebug. The other 2 species are the meadow and sunflower spittlebugs, which can be differentiated based on their size, coloration, host, and spittle mass location. The spittle mass of the two-line spittlebug is typically found on grass or sedge and is usually located near the root, crown, or the lower stem. This is different from both the meadow and the sunflower spittlebug, which spittle mouse found usually on forbs or shrubs and are typically located 4 inches or more above the ground. An additional difference is that the 2-line spittlebug is the largest of the 3 as an adult and is black with orange to reddish stripes. The smallest adult of the 3 is the sunflower, which is mostly brown to black with indistinct markings and is oval shaped. The meadow spittlebug is a little bit bigger than the sunflower, and the adults are various shades and color patterns of gray to brown. The 2-line spittlebug is a major economic pest of turfgrass, forage grass, and sugarcane, and is native to the southeastern United States. It is especially economically significant in Florida and Georgia, where it impacts the landscape trade by damaging golf courses, lawns, and urban landscapes, as it is known to attack all turfgrass species. The 2-line spittlebug undergoes hemimetabolous metamorphosis. The eggs are pale yellow and elongate, and under optimal conditions hatch within 2 to 3 weeks. However, they can overwinter and wait for ideal temperature and humidity conditions. There are 5 nymphal instars, and the nymphs feed in the spittle mass for 34 to 60 days. As adults, they live for about 3 to 4 weeks and begin laying eggs around 2 weeks after emergence. Both the nymphs and adults are polyphagous xylem feeders and can feed on almost all plants that provide fluids to meet their requirements. They've been documented on over 40 different hosts, primarily turf grasses and forage grasses, as well as some ornamentals. They prefer wet and humid climate conditions, and once the eggs hatch, the nymphs begin to seek out suitable feeding sites at the base of the plant. They start to suck the sap, and once it is ingested and digested, they begin to continuously excrete hundreds of white frothy bubbles of feeding waste out of their anus to create the spittle mass. The spittle mass is where they reside as they undergo 5 instars, and it protects them from predators and desiccation. Once they emerge as adults, they no longer produce spittle, but develop the ability to reflex bleed and jump to evade predators. The adults are much less mobile the adults are much more mobile and active than the nymphs and can be seen hopping and flying around in areas of high infestation. When the adults feed, they inject their saliva, which contains amylase, and that interferes with photosynthetic activity. So both life stages inflict damage. This starts with the nymph feeding as they inflict water stress and decrease productivity. However, they do not kill the grass outright. The most significant damage is inflicted by adult feeding, known as chlorosis, which induces phytotoxemia and consequently interferes with photosynthetic activities, leading to decreased nutritional value, palatability, and plant productivity, ultimately killing the grass and promoting the establishment of weeds and lower quality grasses. The 2-line spittlebug was first detected in Kona in 2016. Initial surveys found 2,000 acres of severe damage, resulting in the complete death of grasses that once dominated the pastures, with recovery only occurring sporadically in isolated areas. Like many other invasive pests in Hawaii, it is speculated that this insect was accidentally introduced through the importation of plant materials such as sod or ornamental plants. Livestock ranching is a highly productive and extremely valuable industry on all the major islands. The Hawaiian livestock industry has an estimated value of over $68 million annually, with over 142,000 beef cattle on more than 1,300 ranches. Collectively, they manage 25% of the state landmass. With production still increasing, this industry serves as an economic engine for the state and supports communities of diverse farm workers and Paniolo culture. 60% of the state's cattle are on the Big Island. Additionally, 70% of the beef cattle are supported by sod-forming grasses, including the highly susceptible kikuyu grass. This invasive pest is severely devastating Big Island's cattle industry and is consequently threatening food and job security, the ecosystem services derived from pastures, communities of diverse farm workers, Paniolo culture, and may potentially impact the landscape trade by damaging golf courses, lawns, and urban landscapes. So since February 2018, we've been carrying out multiple surveys to determine the abundance and hosts of the insect. We have 17 100-meter long transects across 4 high-risk Kona ranches that encompass a range of elevations. We have 10 quarter-meter squared plots at each transect where we collect data on plant species composition, nymph abundance, host and location, adult presence and absence, and % live and dead grass cover. So here is an example of the progression of an infestation. On the left, in June 2018, we see an isolated patch of brown dying grass surrounded by healthy live grass. And then
Join
on the
the
right,
meeting.
in January 2019, we see almost complete dieback of the forage grass and an increase in weed infestation. The has rapidly expanded its range and continues to expand at a rate of 35,000 acres per year. By 2020, it was estimated to have increased its range to over 176,000 acres. So the areas sampled encompass a multitude of unique ecosystems and climate zones. So there's a lot of variation in the insect abundance depending on the elevation. So because of that, I split the transects into 4 different elevation groups. And as you can see, there's a lot of variation depending on the year, month, and elevation. So in 2018, the earliest detection was in March, and the latest detection was in November for the low and mid elevation groups, and in December for the high elevation group. There were 2 peaks for the mid elevation group. The first peak was more intense in April, and this was followed by a second, less intense peak in October. That same pattern occurred in the low elevation group as well. And in the high elevation group, we have the highest abundance peaking in May, followed by a second less intense peak in September. And then in 2019, the earliest detection occurred in April, and the latest detection occurred in October for the low and mid elevation groups, and in November for the high elevation. So the high elevation in 2019 had 2 peaks, with the second one being more intense. So the first occurred in June, and the second more intense one occurred in September. For the mid elevation group, there was 1 large peak in June, and the decline slowed down between July and August. In the low elevation group, there was 1 large peak in July. In 2020, we found the earliest detection to be February for the high elevation group and March for the low and mid elevation groups, with the latest detection being in October for the low and mid elevation groups and November in the high elevation. So in the low elevation group, we have 2 peaks, with the second one being more intense in August and the first one in May. The high elevation group, there are also 2 peaks, the second one being a little bit more intense in August, and the mid elevation peaking in August. So overall, the most activity was found between 1,703 to 4,617 feet. And we found little to no activity above 4,681 feet. So the high elevation zone had the highest nymph abundance in 2018 and 2020, and the mid elevation had the highest in 2019. Over the years, they appeared later in the season, and by 2020, they appear earlier. Thus, the overwintering period from about mid-November to March and February could be getting smaller as they adapt to the local climate. Host plant resistance is widely accepted as combat spittlebugs because it is low cost, sustainable, environmentally sound, has a cumulative effect, can be easily incorporated into ranch, and is compatible with other tactics. So because of this, I have carried out 2 different studies in the greenhouse experimenting with host plant resistance. The first study, I looked at the resistance, tolerance, and susceptibility of 6 different grass species to adult spittlebug feeding. So 1 is the most resistant and 6 is the most susceptible. So on the left, you have the control plants, and on the right are the experimental plants. I found Kikuyu to be the most susceptible and Murundu to be the most resistant. So these pictures illustrate this effect really well. As you can see, experimental Kikuyu is almost completely dead compared to the control, whereas the Murundu control and experimental plants have little to no difference. The second study, I assessed Kikuyu response to varying levels of adult infestation. So I infested pots with 5, 10, or 15 adults and found that about 10 adults per square foot appeared to be a significant threshold for Kikuyu survivability to an attack. So as you can see here, the control and the pots infested with 5, 10, and 15 adults. The 10 adults is almost entirely dead, whereas the 5 adults still has some live plant matter left. Currently, we have established 9 field plots across an elevation gradient. Each plot has 32 subplots of 8 grass species. We're trying to look and see if the spittlebugs show a preference for certain grasses over others, and if the resistance tolerance observed in the greenhouse will translate to the field. Additionally, we want to see if the grass varieties will tolerate the highly variable growing conditions on the ranches. And later on, we plan to add a grazing component to determine if heavy grazing can reduce nymph populations by indirectly exposing the preferred nymph microclimate to the elements. The management of the 2-line spittlebug needs to begin with preventing the spread of the pest into new areas on the Big Island and to the other islands. This will necessitate avoiding the movement of sod, potted plants, and live plant materials, carefully cleaning vehicles, trailers, and equipment before traveling, and the landscape industry should be vigilant and check for signs of damage. Early detection is critical, and you should trap and report it if it's detected. The 2-line spittlebug will continue to be a major economic threat, and this will necessitate the development and implementation of an integrated pest management program. This will most likely include using a diversity of resistant and tolerant grasses to replace the Kikuyu grass, proper timing of chemical control, strategic grazing, and potentially the use of natural enemies. So the 2-line spittlebug has expanded its range across a 176,124 acre area and continues to spread at about 35,000 acres per year. When pest densities reach threshold levels, it results in 100% dieback of key pasture grasses, which ultimately promotes the establishment of weeds and lower quality grasses and decreases the likelihood of grass recovery. The critical forages in Hawaii's pastures are highly susceptible. The 2-line spittlebug has rapidly and drastically changed the quality and productivity of pastures due to its preference for key livestock forages that are highly susceptible. The 2-line spittlebug is an existential threat to Hawaii's cattle industry and could fundamentally change the landscape and potential for livestock production to persist. Thus, we must prevent the spread to new areas and to other Hawaiian islands. I'd like to thank my PhD committee, Dr. Mark Wright, Dr. Mark Thorne, Dr. Daniel Peck, Dr. Nick Manukas, and Dr. Peter Follett. I'd also like to thank the research team for hard work in the field plots and greenhouse and field surveys. Additionally, I'd like to thank our research grant support, the Hawaii Department of Agriculture, the Hawaii Invasive Species Council, USDA, ARS, and the Hawaii County Office of Research and Development.
Thank you, Shannon. Questions for Mark and Shannon? Let me check the chat box. There is still questions on Japanese beetle. Oh. Chinese rose beetle. Okay, Dr. Cheng answered some. So questions for Mark from JB.
Hi, yeah, I guess if I'm going to
Yes.
answer this question by JB, the recommendations, yeah, we have some recommendations on management and protocols for addressing early infestations of two-line spittlebug. They're not currently published, but they will be coming out fairly soon. If somebody does spot two-line spittlebug, you know, we encourage them to report that through the 641 pest, 647 pest, or whatever the number is, and or call my office, and we can discuss you know, the location and possible management objectives.
Thanks, Mark. There's another question for Shannon. Have you or others done a financial impact study on correcting the TLSP issue on Hawaii Island? 643 pest. Shannon or Mark? Is there any?
So that study I did assessing kikuyu response to varying levels of adult infestation, we're hoping that will be able to translate to the field and determine how much damage is inflicted per acre, and at what point we need to step in and make management decisions. But other than that, I don't think so, unless Mark, you have more to add to that.
Yeah, actually, you know, we're compiling the economic data. I mean, we some of this information has not been published out there, but we are definitely, because of the ranches that we're working with are having to reduce their stocking rates, and it's not exactly, you know, 100% straightforward. Every location has a different stocking rate. But we're evaluating the
Diana
cost.
was 36 years old when she died in Paris on August 21st.
We're calculating the
Middleton and Meghan Markle often wear
Oh.
jewelry that belonged to Princess
Dr.
Diana.
Klein, is
and
that
they
you?
find small ways to honor her all
that's
the time.
Dr. Klein.
Both have daughters
Dr.
named
Klein.
after her, for example.
What you said.
Charlotte Elizabeth, Diana, and Lilibet Diana.
Okay, no problem, Dr. Klein.
Yeah, so
Yeah.
yeah, the short answer is yes, we're evaluating the economic impacts. We're determining what those threshold values are for the destruction in the pastures and the loss in grazing capacity, and what that economic impact is for the ranches.
Thank you,
Yeah,
Mark.
this is Adams who asked the question. I'm actually looking for, has there been, in addition to that work, which is very important, how much it would cost to fix this? When you're starting to talk about nearly 200,000 acres that's already been affected, plus another 35,000 a year, what are we talking about in terms of the cost of fixing this? That's what I'm asking.
You know, it's going to be pretty substantial. I don't have any numbers for that exactly because it's such a diverse and varied area. You know, I think the long-term solution to the spittlebug issue here, it's not going away. It's going to be around. I think the long-term solution is reseeding pasture with spittlebug resistant varieties of grasses that are being developed in South America. Many of them are available, some are not. They're not yet commercially available. But reseeding is going to be a challenge, and there's some research that needs to be done. There's a few projects underway through the NRCS at looking at different seeding options. And most of these pastures are extremely remote, topographically challenging for equipment. They're rough, they're rocky, they're steep, and you can't get equipment up there. So reseeding is going to be a major effort. Is probably going to have to largely be done through aerial helicopter seeding. That means that the quantity of seed put out is probably going to have to be larger than what you would if you were drilling it. You can't necessarily get out there and prepare or work over the ground post seeding. So as far as each individual ranch is concerned, it's an astronomical amount. What's going to have to happen on the scale for the current area is probably beyond the capacity of anybody to pay for it. That's about the best answer I can give you at the moment. It's going to be an expensive fix.
So some more questions on spittlebug. Is government looking for the Is government looking for the spittlebugs on neighboring island?
So I think the short answer to that is kind of, sort of, Maui, the Department of Ag the Department of Ag is well aware of the spittlebug. I think their employees on each of the islands are alert to that. Maui, we have our ranchers are well informed. That's the next place. Well, we're not expecting it to be there. We hope it won't get there, but that's our one of our biggest concerns is Maui, the introduction there of Maui. So right now it's fairly isolated on the Big Island, and we're hoping that it doesn't get to the other islands, but people are aware of it.
Thank you, Mark. There is some questions on avocado laced bug also. I hope there will be talk on avocado laced bug sometime in the next mini conferences. For now, we will go ahead with the laurel wilt and the two-line spittlebug. And what else? I'm checking my chat box. So this is a question for Mark Hughes. Can you speak in general to wilt, forestry disease early detection in Hawaii? What is happening to actively looking for tree diseases like laurel wilt if it was able to get past border inspection?
Okay, can you hear me okay?
Yes.
Okay. And I'm hoping maybe JB Friday and Rob Hoff can add to this, but I believe our detection system is really reliant on the collaborative network of the state, university, and federal organizations like the HISCs and extension agents to be able to kind of report any unusual findings. And also a lot of these also in like rapid ohia death work is doing a lot of surveying and scouting. So that could be another potential option for reporting new and unusual disease occurrences. I'm not sure of any potential organized program to scout for, brand new forest diseases, but I believe that's kind of how we do it at the moment.
Yeah, Mark, if I could just add to that, it's the private landowners. It was private landowners who brought rapid ohia death to our attention. It wasn't any of the agencies, but sure, it's forestry and wildlife spends a lot of time observing the forests, the invasive species committees on every island, and the watershed partnerships, because they are groups that are out working in these forested areas. But the private landowners also provide a key part of that, and monitoring and just seeing what's going on and bringing it to our attention.
Thanks, JB.
Thank you. So I have one concern, just a quick question. If we do not have a red bay ambrosia beetle, can we assume we will not have a laurel wilt here in Hawaii?
That's a good question because it's hard to say, but I would think that you need that primary vector, the red bay ambrosia beetle, to come first and start infecting trees. You know, those other ambrosia beetles that I mentioned, a lot of them already occur in Hawaii. It's just that they're not able to co-infest an infected red bay tree to pick up the fungal spores. So a lot of those other species that I talked about, those are worldwide. Some of them are worldwide distribution. You know, they're hyper generalists. So I think the real red flag would be, and I don't want to scare people when you say, oh, if you find these other species here, you have laurel wilt. They have to, those other ambrosia beetle species would have to be already attacking a laurel wilt killed tree. So the red flag would be if you find the red bay ambrosia beetle here, because that association with the beetle and the fungus are so closely woven that if you find the beetle, you probably have the fungus here too. But again, even if like the disease triangle, even if you have the beetle here, you still got to have it interact with the host. I think those are things that bode well, and, you know, in Hawaii, just because there's not a lot of forest lauraceae here. But again, you know, before the disease got to the avocado production area in Miami-Dade County, there were avocado trees dying all the way down, mostly people's backyard avocado trees. So, you know, they could still, the red bay ambrosia beetle, it's assumed that that mortality was occurring by the red bay ambrosia beetle. Maybe it was these other hosts picking it up. But I think the red flag, the real red flag is arrival of the red bay ambrosia beetle.
Thanks, Mark. Just wanted to check again, the laurel wilt is only on Miami-Dade County in Florida, not in anywhere else?
No, so the disease is, it's all throughout the southeastern United States. I don't know, I could pull up the distribution map, but to clarify, it's only affecting commercial avocado production in Miami-Dade, Florida. So there's big production areas in California. You know, Florida does like 10% of the avocado production in the United States, and California does like 90%. I'm not sure where Hawaii fits in there. I'm sure there's a few percentage points for Hawaii too, but it's not in California and it's not in Mexico. So that's what I was saying. That's the only production area where it lives.
Okay, thank you. Yeah. So next, it's on spittlebug. Will the spittlebug attack guinea grass? Shannon or Mark?
Shannon, do you, I guess, yeah, we've seen it on guinea grass. We've seen it on a very wide variety of grasses. I think the biggest question is not whether it'll attack the grass or not, it's whether or not the grass can tolerate or resist the damage that the adult spittlebug can inflict. And that's a lot of the work that Shannon's been doing, is looking at the different varieties of grasses. Now, last year she started on six grasses that we determined were critical to look at. She's doing trials on another 8 grasses this year. And, you know, because the pest is seasonal, we have a very short window to run all of these trials. But the short answer is yes, it will attack guinea grass. We've seen it on the guinea grass, but mostly the guinea grass seems to be a little bit tolerant. It doesn't necessarily kill it out. But then again, we haven't seen the critical threshold numbers, densities in those areas that guinea grass grows. As you saw from Shannon's data, the pest is strongest in the mid elevations for above 2,000 feet and below 4,000 feet. You know, we get below 2,000 feet and the environments get a little bit less hospitable to the pest, and above that 4,500, 4,600 foot elevation, the environment is seems to be a little bit less inviting to the pest as well. Although we've seen spittle bugs above that elevation, they don't seem to be causing any kind of damage, and it may just be that the populations haven't made it up there in that density yet. There could be a lot of factors.
Thanks, Mark. The 2 peaks in the mid elevation means 2 generations per year.
That's 2 generations per season. And that,
Season, okay.
that's the season usually starts as Shannon showed in her data, roughly between March and November. That's sort of the season, the main peak of the season. But we are starting to pick up some drift, I guess, in that sense that they're starting to show up more frequently, even in the winter months, which is a little bit concerning.
Okay, thank you. So it's already 12:04. I will just take a few more minutes, maybe 10 more minutes to go through these questions in the chat box. So there is one question for laurel wilt, I think. What tree spacing discourages root grafts? Mark.
Oh, what trees? That's a great question. I think root grafting is, I don't know what spacing necessarily. I think a lot of it has to do with the substrate that the trees are planted in. Softer substrates allow for roots to go further, like in oak wilt in Texas. The root grafting can happen pretty far distances. One of the questions originally brought up with Rapidohea death was it occurring through root grafts? And if you think of those trees growing in certain sites, in this volcanic substance, we think it's less likely that the roots are making contact, because root grafting is only with the thicker woody roots. It's not fine feeder roots. So I don't know in particular an exact distance. Obviously, the further the better, but it's hard to say for sure.
So Mark, it looks like the other general ambrosia beetles also can transmit the laurel wilt, yeah? As you saw.
It seems to only be occurring in avocado groves, and only again in trees that they're only picking up the fungus from trees already infected with floral wilt.
So you have one slide that shows red bay, which is infected with the laurel wilt first, and the other generalist ambrosia beetle will attack. Then they will move to avocado group, yeah?
That's the thought, yeah.
Oh,
they have to pick it up
mm-hmm.
from an infected tree. These other species, Rafflesia loricola, is not the fungal symbiont.
Okay.
It's not generally associated with those beetles. The pathogen is generally the natural symbiont is the red bay ambrosia beetle. So as long as we don't have the red bay ambrosia beetle and laurel wilt here, I think the chances, I don't see any real chances of laurel wilt becoming established, because the pathogen isn't here in the first place. Even if those other species of beetles are here, the pathogen isn't.
Okay, thanks, Mark. There's one question from related to landscape industry. I think it's addressed by Dr. Cheng. Okay. So again, question on laurel wilt. Above and beyond the Lauraceae family, does laurel wilt disease infect any other plants in the laurels order?
As of right now, it's only been the Lauraceae that have been infected. Even in like Asia, where they found laurel wilt, it's only in avocado trees. And there's been some studies done in Florida looking at artificial inoculations of other host species beyond the Lauraceae, and none of them have shown to be symptomatic. So as of right now, it seems like it's only within the plant family Lauraceae. And it's really the North American members. A lot of these Asian members of the Lauraceae seem to be a lot more resistant to the fungus, which kind of suggests that coevolution in the natural range between host and pathogen It's only these new hosts in America that seem to be hyper susceptible.
Thanks. So any other question, last minute questions for Mark and Shannon or Mark Thorne? So if not, you can also